Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. Delighted to see you, David and Yuan. As you know, we're having conversations with participants and speakers for the Cancer Neuroscience in next February. Amazingly, it's coming up sooner. So delighted that we have this chance to chat with you. And what we've been wanting to do is really just sort of get to know the speakers and a little bit about their backgrounds and the work they're doing, their vision for the future of cancer neuroscience. And so maybe a good place to begin really is to ask each of you, maybe David, you could go first, to give us a little bit of background on how you in particular ended up in St. Louis, and Yuan, you in Texas, and how you came to begin and continue doing research in neurofibromatosis and cancer. So maybe David go first, and then Yuan.
Absolutely. So my journey began as a physician scientist. As a physician, I care for folks with neurofibromatosis, which is a cancer predisposition syndrome, and children and adults develop tumors that are always intimately associated with nerves in their brain and their body. As a neurologist, I take care of kids with NF, but I view them as chronic diseases. And so I believe that the rules that govern normal functioning of the brain should also be the same sort of rules that tumors try to usurp when they grow. So this idea of tumors as a chronic disease and intimately associated with the nervous system really drove my interest in cancer neuroscience.
Yeah, so I started as a basic scientist. So my PhD was trained in looking at how proteins are trafficked within neurons in the uptake pathway. So after I graduated, I was looking for a lab that I can use the things I learned during my doctoral training and expand a little bit to a different field. So that's when I found David, who studies NF1 and uptake pathway glioma. So it's a perfect fit for my background. So I joined David's lab, and since then, I've been just fascinated by NF1, and because there are so many aspects to look at. There are the cancer depositions, and then the neurological issues with NF1. So that's two major areas we're spending our efforts in research.
It's interesting, so David, you were mentioning, predisposition. And so I guess, what is it, maybe 10% or so of people with NF1 develop malignancies? And so there's a lot of heterogeneity. Could you both speak to that heterogeneity and why that is? Why is it so complicated, and why do the majority of people with this mutation not develop malignancy? Why do some do? And just sort of open up that conversation.
So that is one of the major failures that we as physicians encounter when talking to families. Is we can make a diagnosis pretty easily, but then they ask us the really important question, which is, okay, so what's going to happen to my child? And that, for us, is something that we can't answer any better than we could 10 or 15 years ago. Mm-hmm. Then they ask one more question, which is even more vexing, which is, okay, so my kid develops a brain tumor. How are you going to treat that brain tumor that arises in NF? And we say, well, we borrow things from other cancers that, might work. And so I think we really do our families a disservice right now by not being able to predict what will happen and by not having therapies that are targeted to their disease. These become the driving forces behind research because we need to be able to define what contributes to heterogeneity. Is it the mutation that you're born with? Is it the environment, the cells that interact with the tumors? Is it something that's in your gut microbiome? Is it something that you're exposed to? Because all these things together probably contribute in small ways to confer risk for brain tumors or other kinds of cancers in NF1.
I mean, it's very interesting because, like BRCA, one, there's a much higher penetrance of cancers. So, some of these factors, these second hit factors that I guess are contributors to ultimate outcome, is an extraordinary opportunity and definitely a call to action for sure. You
It's
want further thoughts on this heterogeneity and what factors might contribute? David mentioned the microbiome and potentially the exposome. Are there other second hit kinds of factors or environments that you think are gaps in our understanding that might contribute to ultimately the outcomes for NF1 mutations leading to malignancy?
Yeah, when I generally think about this question, I make it one is cell and gene intrinsic, the other one is external from the environment. And like I think we you touch a little bit on the second hit, right? That that's a question we're still exploring right now. Like which cells have the second hit, and how much the second hit influence the symptoms. Of course, second hit is important for tumorigenesis, but we also think maybe other symptoms we're seeing are also related to the second hit, but it's not very sorted out yet. And just based on analysis on non-malignant or non-neoplastic tissues in postmortem patients, samples, we can see some that like it was published recently that there are second hit NF1 loss in some normal looking tissues. So that could be the contributor, just where the second hit happens, which cell types are affected. Um, Externally, like I think following the microbiome, which means diet could have a profound effect. Dr. Nicole Brazier, her research showed that diet will affect tumorigenesis for NF1 optic glioma. So yeah, that's potentially can link to microbiome or some other mechanisms, something that's worth looking into for sure.
asthma creates a population of T lymphocytes in the lung that traffic to the brain and interrupt the normal communication between T cells and monocytes. And so you break that circuit that supports tumor growth. There are other ways that uh, your exposome or your microbiome or either other cells work by converging on the circuits that maintain the tumors. So the microbiome uh, acts by converging on tumor-associated monocytes. The work that Yuwin's been involved in has really been focused on how mutations might affect neurons. And then there are other factors, including race and ethnicity, obesity, et cetera, that we're still sort of working our way through as a collective in understanding the etiologies.
Yeah, I think it's interesting the idea of, you know, crosstalk between cells, leukocytes and neurons, neurons and the tumors, and that's an important thematic in the Cancer Neuroscience Symposium. Well, I think we'll have quite a few persons speaking to this issue. But I guess there's something about NF1 mutations where the hyperexcitability of those neurons has been, I guess, linked to behavioral memory learning. And I wonder how many people with NF1 display these sort of cognitive phenotypes versus malignant tumorigenesis. So does one precede the other? Could you sort of speak, maybe both of you speak to sort of the behavioral sort of cognitive phenotype that is sometimes described in people with NF1?
I'm happy to give a stab. You and I just came back from a focused meeting at Cold Spring Harbor at the Banbury Center that was really directed at understanding what are the uh, cognitive and behavioral problems in folks with NF1. Uh, there are clearly many more patients affected by cognitive and behavioral problems than there are by tumors.
Yeah.
The relationship still needs to be worked out about whether or not autism is protective against brain tumors or attention deficit might be uh, associated with greater incidence of brain tumors. That still needs to be sorted out. Uh, but it is fascinating because both of these are networks involving multiple different cell types, both tumors and learning and cognition, uh, and they are both heavily influenced by factors outside of the brain. And so,
Yeah.
it is very possible that the circuits that are established by both of them are influenced by NF1 in different ways. One might promote learning and behavior problems, and the other might promote or inhibit tumor genesis.
Yeah, I want to probably add our recent publication, which is where we are deleting or mutating NF1 in the oligodendrocyte precursor cells. So they you can think it both ways. So they make oligodendrocytes, and which are important for myelination, neurological function. And then these progenitor cells are also the putative cell of origin for gliomas. So when we knock out NF1, these precursor cells become preneoplastic. But our study focused on the neuron communication part. We found that without NF1, these OPCs, they don't really respond to neuronal activity. And for the wild type scenario, it's really important for OPCs to talk to neurons because that's how they decide whether they should differentiate, whether they should perform adaptive, well, they should support adaptive myelination. And that's an important process for learning. And NF1 loss in these OPCs that impairs motor learning. So I think we have some link between pre-neoplasia and neurological deficits, but we still need to sort it out because we haven't done a lot of different behavioral assays to look into, for example, cognition.
Yeah, it's fascinating. I wonder to what extent there are early behavioral biomarkers, with that may be linked to this demyelination process, driven by the oligodendrocytes lacking neurofibromin. Do you think that biomarker development at a more holistic level, looking at persons for whom there's clear risk through genetic analysis, to grade potential trajectories for these persons? Maybe there's the early cues of behavioral phenotypes that suggest that they're going to have a more severe neurofibromatosis associated cognitive outcome versus a more mild case, something perhaps similar to like what you see in sickle cell, where there's a wide variety of severities. So just in terms of the patient trajectories, are there strata? Are there different trajectories, and are most of them involving early evidence of demyelination and associated memory and learning, phenotypes, and then a certain subset of those go on to develop gliomas or cancers, or is that all still a very big question about the different trajectories that define severity?
So the time frame that each of these clinical problems arises is overlapping. So it's hard to draw cause and effect just because the time frame is short. So optic gliomas are a disease of young children, typically between the ages of 2 and 6.
Yeah.
You've got autism, which really doesn't begin to become clinically evident until after 18 months and closer to 3 or 4 years of age. You've got learning problems that really are not uncovered often until kids are well into elementary school. But the earliest sign of this may be something that is linked to brain tumor genesis, maybe defects in myelination, maybe defects in something else. That's the root cause of all of those. Just the temporal course is a little bit different.
How often I'm just thinking about, you mentioned kids. How often is it germline versus somatic mutations are there differences in the clinical sequela, I guess, or the biological sequela if it's a somatic NF1 mutation versus an inherited autosomal? Or do they present very similarly? Or, maybe a kid that's 4 or 5, then they develop an NF1 mutation in the somatic tissue, and that is that something that contributes to the heterogeneity, whether it's germline or later in life, even if it's early in life?
I mean, typically everybody with NF1 is born with the mutation in their germline. There are some that develop the mutation during development. It's only one segment or portion of their body might be affected. Other than the sheer amount of real estate that's involved, those two seem very overlapping. And even if you looked at the range and types of mutations in, say, the cancer genome atlas, in the NF1 gene in other cancers, and compare those with those that occur as germline mutations in NF1, they both run the spectrum.
Yeah.
I think for the tumor side, David's group has modeled a few germline mutations, patient-derived germline mutations, in terms of their what's the tendency to form optic glioma, and it's depending on the germline mutation. We've just started trying to model that for the cognitive neurological deficits in mice. So it is ongoing. Clinically, there are more than 3,000 NF1 mutations found. Not a lot of them have the genotype-phenotype correlation, but there are some that shows a correlation. Some have a correlation with neurological deficits. So that's what we're looking at. Are those mutations, if we put them in the mice, do we also get more profound impairment in those behavioral assays?
Are they all loss of function, or are some of them like reduced suppression of RAS? I mean, how variable are these mutations in terms of overall RAS signaling effects? Because I guess that's one of the early downstream targets.
It's a great question. You want to take that? I mean, I'm happy to.
Oh no, I was just gonna say the data you have.
So I think the data's pretty clear that they're all loss of function. There's no real evidence for gain of function mutations.
Yeah.
But one of the vexing things that we've all struggled with, Yuen's lab included, has been all of them result in increased RAS activity.
Mm-hmm.
So RAS can't be the entire answer. It is part of the answer, but there must be other things that this very large protein does.
Yeah.
And those are going to be very fruitful investigations in Yuen's lab and our lab and in other labs.
Yeah, I think it's very, very, very interesting. I think I remember reading somewhere that there is also an effect on the mTOR pathway. So not only RAS signaling is elevated, but mTOR is also elevated, and mTOR has very, very broad effects in terms of growth and even lifespan. Uh, and so it's perhaps the pleiotropic targets that neurofibromin. It's Are there proteomic? I guess the signaling pathways across mTOR, across RAS, are well described for NF1, and so perhaps the severity and the phenotypes boil down to omics. And I guess my question would be, given that we understand that cancers are so heterogeneous and require recognizing that heterogeneity needs to be measured, do you think techniques like spatial transcriptomics and single cell, uh, you know, um, phenotyping in tumor tissues and tumor biopsies is going to provide some insights? What are your thoughts on that?
Yeah, so the recent study, what we found is that NF1 heterozygosity leads to OPC hyperdensities in the brain, and they look very much like preneoplastic lesions, potentially that can form gliomas. But we don't know molecularly what's going on within those hyperdensities. And that's where the spatial transcriptomics is going to be very helpful. And especially nowadays, we can look at single cell level and spatially. So that's something we really want to try. And yeah, so I think the technique would be really helpful in that
Do you
sense.
think that the differences in those regions are going to break down into the amount of infiltration of leukocytes, the effectiveness, maybe the senescence signatures and how strong they are? What are your guesses as to what might make the difference between focal tumors and some of these phenotypic differences you're seeing?
So our first guess is actually those hyperdensities or spontaneous loss of heterozygosity.
Mm,
Um, but like NF1 researchers knows that we don't have a great antibody for staining or RNA probes to stain. So I'm imagining using spatial transcriptomics when we get to single level. Hopefully, we can detect the level of NF1 in
mhm,
those cells, in those patches.
yeah.
Yeah, but looking at the other, we have looked at microglia astrocytes within those hyperdensities. They look pretty normal.
Hmm.
So in terms of whether other cell types are affected, it's still a question mark.
You say they look pretty normal, but all of these cells have reduced NF1 expression, right? So because that's genetically wired into your study system. So I'm just thinking about, are there different thresholds for like T cell function, glial function, senescence induction that rely on RAS signaling, and so these different thresholds may fluctuate randomly due to the local environments, and that susceptibility to the triumvirate of the tumor, the neurons, and the leukocytes. Uh, if there's a perfect storm, then things take off in the bad way. So I feel like that there's it's an extraordinary paradigm shift here, because rather than things being very canonically A to B to C, we have to sort of incorporate this idea that there's a certain amount of fluctuation and thresholds across interacting cell types that ultimately lead to this, as you say, this spontaneous transition into a malignant phenotype. And so I think this is something that we're super excited to hear more about in the conference, in February, and actually, I guess beyond that. Um, is the presentation of NF1, does it differ in adults? So, David, you mentioned a lot of these gliomas in kids, and there are different cancers associated with NF1, and so how is is the pathobiology of NF1 associated malignancy in kids different, and if so, how is it different than, say, when it occurs in adults?
So I think that's where thinking about tumors of the brain as a neuroscientist or a neurologist is helpful, because it's all about context. If you look at where these tumors arise in children, they're in different locations than they are in adults. They probably have different cells of origin, even within the optic pathway, which includes the nerve that connects the brain to the eye, to the nerves that actually go to the back of the brain. Those tumors that arise in one region may have a completely different cell of origin and clearly have different clinical biology. So those in the optic nerve, for the most part, tend to be reasonably responsive to therapy. Some even involute spontaneously. Those that are in the brain, those optic pathway gliomas are very difficult to treat, often occur in much younger kids, and are associated with significant morbidity.
Yeah.
So I think as we begin to dissect the heterogeneity, we need to stop lumping everything as a brain tumor in NF1. It depends on when it occurs, where it occurs, and who's in the village. Because these are circuits, just like the brain is wired as a circuit. Circuits have both individual nodal information, like what each cell does, but they have a spatial context, because if they're not arrayed in a particular way, you're not going to have a functioning circuit. So that's where I think having the deep dive that some single cell nuke or single nuke sequencing will provide, and the spatial component, gives you a great idea of how those circuits function, how they're arranged, and maybe how you can interrupt them.
Yeah, I think it's fascinating. There's something happening at the tissue level in the tumor, and it's complex, and it's going to require some of these surveillance tools that look at the different tissue components and contributors, and I guess the paracrine factors that interact in that context. But I guess I'm thinking about people walking around at risk, and are there liquid biopsy tools that you think might provide some clues to the heterogeneity that's happening perhaps in an optically related tumor or a glioma that might serve as proxies for progression, because you won't be able to do spatial transcriptomics in a 4-year-old kid, but maybe you can infer that something's going on from maybe the circulating proteome, or maybe even metabolites. Are there thoughts on developing some of these based on what you know, and you guys are the experts on sort of these paracrine interactions and various cytokines that I think are being secreted and delivering and recruiting cells? Are any of those useful as circulating biomarkers? What are your thoughts on being able to surveil the peripheral blood to get a sense for categorizing this heterogeneity that seems to be a formidable challenge?
So in my mind, I think the approach has to cast a wider net. And so the way that we're approaching it, our colleagues are approaching it, is by trying to understand in an unbiased way what are the factors that contribute. How do they contribute both singly and as in combination?
Yeah.
It is, part of that might be the germline mutation. Part of that might be, what your body weight is. Part of it may be something else. Array these factors, bring in laboratory data, bring in genetic data, bring in maybe one day circulating tumor DNA, so that we get a bigger and perhaps less biased view of what really constitutes accurate risk factors. Because right now, low-grade brain tumors are pretty poor at shedding their DNA into the circulation. So, we would have to do lumbar punctures. That's associated with, some degree of struggle. So I think a good way to think about this might be really to deconvolve all of the factors that could contribute to the pathogenesis of the disease,
Yeah.
and then use those as risk assessment tools.
I think it's fascinating, and I think you mentioned all of the factors. I think about the mouse and its behavior, and how can you multimodally, not just spatial transcriptomics, but the behavior of the mouse in the cage, and maybe even stress tests on that mouse and sort of revealing a hidden phenotype that precedes perhaps tumorigenesis, and maybe this sort of moving away from the detail to see the bigger picture and how there might be. I don't know, Yuan, you've been looking at a lot of these you were mentioning some behavioral phenotypes. Have you noticed anything, prior to obvious tumors and in the optic nerve or elsewhere in those mice that might offer some clues to early, maybe functional biomarkers that are not necessarily circulating biomarkers, but more phenotypic, larger scale behavior patterns?
That's a really interesting question, which also means we need to track the genetic engineer mice that develop the tumor by doing behavioral assays, like before tumor formation.
Yes.
Right? So our current NF1 optic glioma mouse model, it has a NF1 deletion in neural stem cells. So that's of course going to contribute to the tumor genesis, but because it's also neural stem cells, it's also going to affect their progenitors. So the mouse model itself is hard to distinguish because of the way they were engineered. So if we can find another model that we can clearly separate, like this is because of the tumor, this is before the tumor, but still only the germline mutation is in there. That would do a better job, I think, which we're currently trying. So we can potentially do implantation. Yeah, but those are low grades, so we need to carefully characterize their timeline of tumor formation. So everyone who works on tumor initiation had this struggle of when do we define the tumor actually forms in the mice. So we need to do a longitudinal study to begin with.
Yeah, no, it's fascinating, and I think it's an extraordinary challenge, but it's absolutely essential that we change the way we think about cancer and tumors to being a progression of steps that are increasingly complex, and even to the level of the person. So I guess we're looking forward to hearing more about what you guys are up to. In February, we look forward to seeing you there, and and congratulations on your great work. It's such important work. And thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.